Now, the third macromolecule we mentioned were proteins, and proteins account for about 50% of the dry mass of most cells. So there's a ton of proteins in living organisms, and they do so many functions, and they're incredibly important to our survival. But, like the second bullet says, and I just mentioned, they are instrumental in almost everything organisms do. And just some different functions that are listed here. It says they include speeding up chemical reactions, structural support, storage, transport, cellular communications, movement, defense against foreign substances. I'm not going to ask you to repeat that list by any means. I just want you to be aware that proteins do an immense amount of work. And again, this is just another table with that's kind of illustrating all the functions proteins do for us. Now, when we talk about proteins, we have to mention something called enzymes. And enzymes are a type of protein. And it says here, enzymes are important types of proteins that regulate metabolism due to their catalytic nature. So throughout the class, we're going to talk about different types of enzymes and why they're important and how they help with different types of chemical reactions. But here it says they have a catalytic nature. So let's define a catalyst. And if you've taken chemistry, you've probably heard that word before. But a catalyst is a chemical agent that speeds up chemical reactions within a cell without actually being consumed by the reaction. So enzymes are a type of catalyst that our body can use to speed up chemical reactions. Now, polypeptides. That might sound a little bit familiar, but different. The first macromolecule we talked about were carbohydrates, and we mentioned polysaccharides. Well, instead of sugars, now we're talking about proteins. And so if we're talking about many proteins that are bonded together, we're going to call those polypeptides. So it says proteins are all polymers made from the same set of 20 amino acids. So that's a great test question. If I was to ask you, what is the individual building block or what are the monomers of proteins, you would tell me amino acids. And you would tell me that there's only 20 possible amino acids or 20 possible building blocks that can make up a polypeptide. Now, a protein consists of one or more polypeptides that are folded and coiled into specific conformations that we're going to talk about. Well, amino acids are organic molecules that have both a carboxyl and an amino group. And we'll talk about that more in just a minute. But it says here at the center of an amino acid, we have this kind of asymmetric carbon atom that we call the alpha carbon. So we're just kind of describing what an amino acid is at this point. And it says here it has 4 different partners. Okay, we have an amino group, we have a carboxyl group, a hydrogen atom, and then we have this group that they're calling a variable group. And they're going to symbolize that by an R, or by the letter R. And this R is also sometimes called the side chain or the R group. And I really want you to remember that amino acids contain this R group because all amino acids are exactly the same except for this side chain that they're referring to as the R group. So this R group kind of distinguishes individual amino acids from one another. So it differs with each amino acid. So here's an illustration of everything we just talked about. We have that central carbon within our amino acid, which again they call that an alpha carbon, and it's attached to a hydrogen. On the left side we have this amino group. On the right side we have a carboxylic acid group. And then on top we have that R group. And remember, there's 20 types of amino acids. They all have the same structure except for that R group is going to be different in all of the 20 amino acids. And this picture, don't get scared, it's just here to show you the different amino acids, and it allows you to kind of see how they all indeed have that same structure except for these boxes that are highlighted with white. And those are the R groups I was describing. So you can see that when you compare the different amino acids, that R group is going to be the variable factor. All right. Now, we talked about how we build polymers and we link monomers together, but how specifically do we do that in proteins? It says here, when two amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of the other, an enzyme can cause them to join by catalyzing a dehydration reaction. And that's familiar to us, but this time we're talking about proteins. So let's give a specific name to these bonds or these links that form between protein monomers or protein amino acids. It says here the resulting covalent bond is called a peptide bond. Okay, so in carbohydrates, if you remember, we said those linkages that exist between the individual monomers of carbohydrates were called glycosidic linkages. If we're talking about proteins, we call them peptide bonds. And again, they're formed because of that loss of a water molecule, because of a dehydration reaction. And what happens is we get this process repeating over and over again, and we get the formation of a polypeptide. And polypeptides can range in length greatly from just a few monomers to over 1,000. And just to remind you, I have kind of a visual here to show you how this peptide bond is formed. So on top we have our individual building blocks. Okay, on the top left we have two that are side by side, and they've already established a link between them. So they already have a peptide bond connecting the two. But we want to go ahead and attach that kind of lone purple guy on top. So we're going to utilize a dehydration reaction. In the process, we're losing a water molecule. And then at the end, we're going to have a polypeptide that consists of 3 different amino acid groups, all linked by peptide bonds. So polypeptides or the structure and shape of polypeptides have been studied a lot, and they were first determined or kind of first studied by chemical means, but now